Welcome. This is Anna here, and we are now going to work on part 6 of the neurophysiology chapter. This one we're going to do a short compare and contrast of local potentials versus action potentials. So this is basically a type up of notes that I have that I did at the end of the previous presentation, where I talked about the things that you can do to change membrane potential, your definitions of depolarization, hyperpolarization, and then right here, the important part, the major difference between a graded potential and an action potential. Okay, so these are note slides. I'm not going to talk about them. Instead, we're going to go to the whiteboard and we're going to start talking about that. All right, so what is a local potential or a graded potential? Graded is talking more about how big a signal is. Okay, if a whole lot of sodium comes in, it'll be big. If not a lot of sodium comes in, it'll be little. Local is conveying this idea that it's a short distance thing. All right, it stays in one little spot. So if I'm drawing a neuron here, all right, so there's my cell body, here are my little dendrites, here's my axon, the axonal terminals. A local potential is going to be like here. Maybe it'll be like this. Maybe it'll be like this, okay? It doesn't get very far. It stays local. Now, if I want to generate an action potential, I need to get the signal to there where I have voltage gates. So it needs to have enough sodium coming in that I can start a signal here and it's got enough sodium to get to here. So that would be a big grade. This over here is a little grade. It fizzled out before it got to the trigger zone. All right, and remember the trigger zone is in the axon hillock. All right, so local potentials are short distance. Graded potentials are either small or big, okay? But both of these terms are used interchangeably. So we will say a graded potential and mean a local potential, or we can say a local potential and we mean a graded potential, okay? They're the same thing. So what stimuli are going to be used to generate a local potential, okay? The most common one that we are familiar with is going to be chemicals. These are your neurotransmitters. But it's not the only thing that can start a graded potential. Light energy can as well. Heat, voltage change, which we've already talked about a little bit, mechanical change. These are all different types of stimuli that can start a graded potential on a neuron, okay? So what are our steps? So first, you need to open the gate. And this could be a chemical gate, or it could be a mechanical gate, or it could be a photoreceptor gate, a lot of different things, so etc. There are other types of gates. These are the two most common that we're going to be talking about, okay? Once the gate opens, sodium flows into the cell, okay? That is going to cause depolarization. This depolarization, or actually it can cause either depolarization or hyperpolarization. This depolarization or hyperpolarization is the polarity change to the resting membrane potential. This is your graded potential, also called local potential. It's that change in polarity right there, okay? So let's summarize the properties of a graded potential that make it special and different from an action potential, okay? So we're going to do GP for graded. We're just going to make a table over here, and we're going to do AP for action potential, okay? Graded potentials, the word graded, all right, means small or big, okay? And this is completely dependent on how much sodium enters, okay? Action potentials, always the same size. All right, they are always all or nothing. If the voltage gate opens, you have an action potential and lots of sodium enters the cell. Okay? Graded potentials are also called we also use the word decremental to describe graded potentials, okay? Basically, that means the further you get from the stimulus, the weaker the signal, okay? So remember, the stimulus is where you open the gate. So the further you get away from the gate, the further the sodium has to go. So eventually the sodium can't walk any further. It gets tired and it just fizzles out. So it just stops, okay? Graded potentials are also reversible, okay? So if the stimulus stops, it repolarizes. Or if there's a counter signal. So if I'm using a neurotransmitter that depolarizes a membrane, but then I use a different one in a different gate that hyperpolarizes it, that can reverse the depolarization, okay? Now, action potentials never reverse. Once they start, they do not stop. All or nothing. Okay? Now, graded potentials can be excitatory or inhibitory. Okay, so I've already mentioned this, but I've used different words. It can depolarize or it can hyperpolarize. Okay? Action potentials depolarize. So they are considered excitatory. Okay? And the other big thing to remember, actually, let's change color. All action potentials are started by local potentials. You can't start an action potential unless you open a gate and you try doing a local potential. So if the local potential reaches threshold, then opens voltage gates and you get an action potential. Okay, let's look at the next slide. All right, let's kind of look at some of these ideas, okay? So this local or graded potential is going to be triggered when some kind of gated ion channel opens. And this could be a chemical gate, it could be a mechanical gate, okay? It's just something that opens. In the case of like some other things like calcium, it might be a voltage gate as well, but it's not a voltage gate related to an action potential, okay? Now, we've got some other definitions right here, like generator potential, okay? The generator potential is just talking about how you have some kind of receptor that is stimulated by an energy source. So it could be heat, light, something else, okay? Or you can have a postsynaptic potential. And this is where a neurotransmitter starts the graded potential, okay? So generator potential is going to be like special senses, okay? Whereas postsynaptic potential is going to be neurotransmitters, okay? That might be a way to keep them straight, okay? So what opens the gate in chemical muscles and skeletal muscles, I guess I just gave it away, that is acetylcholine, which is a neurotransmitter. All right. All right. Next, I want to look at this picture a little bit more closely, and I think this is a really clever picture. So if you look right here, you can see I've got neuron number 2 here and neuron number 1 here, and this box right here is being blown up over there, so that this is neuron number 2 and this is neuron number 1. Okay? And what I have is my stimulus secreting a neurotransmitter. It could be acetylcholine, or it could be dopamine, or it could be epinephrine, or it could be GABA, or all kinds of stuff. But whatever happens, it opens up this chemical gate which allows sodium to come in. Now we've just got the gates right here. There are no voltage gates nearby. Okay? And basically the sodium is going to diffuse out from its source. But the further it gets away, the concentration goes down. And you're only going to have sodium coming in until this gate snaps shut. And so eventually here, you can see in red, it's representing that there is a strong signal. And then over here in this pale blue, it's representing weak depolarization. Okay? So this is that idea that at the source of the stimulus, the signal is strong, and as you go away from the stimulus, the signal becomes weak. Okay? Now this right here, this graph is showing you a similar thing. So we've got here, this is the stimulus source. So from here, this is going away in different directions. This is the strength in millivolts. So this is showing you that at the source right here, you have the highest strength. And then as you go in either direction, the further out you go, the weaker the signal becomes. Okay? I think this is a really clever picture the way they did this. I like this one. Let's look at the next picture. All right, and this picture, we're showing a sub-threshold graded potential, which means it will never generate an action potential. Over here, this is a graded potential that will be able to generate an action potential. Okay, let's look at why. Here, we're looking at neuron 1 and neuron 2. Neuron 1 has a stimulus that's coming down. It secretes the neurotransmitter. I'm just going to use acetylcholine because it's short to abbreviate. This is the stimulus source. Okay. Then as we go along, you can see the signal gets weaker and weaker and weaker before it gets to this area. So this is the trigger zone. This is the zone where I have voltage gates. But you can see, let me erase that so you can see it a little bit better, that right there, the signal died right before it got to the trigger zone where I have voltage gates. So no voltage gates are opened, no action potential happens. Let's look at the graph. So here we have resting membrane potential. So this is before the stimulus hits. This is right here where the stimulus starts. It comes in, and at the stimulus source, right here, you can see, oh my God, it got up to -40. It totally could have started an action potential, but no voltage gates nearby. It doesn't matter, all right? Now we get to the middle right here, and we're going to look at its graph. So here's the stimulus. I've got -70. I get up to -55. Theoretically, I might be able to generate an action potential, but again, no voltage gates. And then I get to right here where I've got voltage gates. And look, here's my stimulus source. This is all the time. By the time I get to the area where I've got voltage gates, it's no longer at negative 55. I cannot generate an action potential. So, fizzles out. No action potential is created. Now, let's look at C. I've got neuron number 1, neuron number 2. I've got my stimulus source. It opens up the gates. Now, notice what they've done right here. See the size of that triangle versus the size of that triangle? They're bigger. So they're trying to represent that this is more sodium came in. So it was a stronger stimulus from the start. And then right here, if we go look at the graph, you will see here's the stimulus source. Yeah, it got up to where it could have generated an action potential because it passed threshold, but no voltage gates, so it doesn't really matter. Now we get to the middle of the cell body, and we're going to go look at the graph. We're here at negative 70. We get up to here. Yes, we pass threshold. I could generate an action potential, but no voltage gates. Now I'm going to get down to here. This is my trigger zone. There are voltage gates. So we're going to come over here and look at the graph. Here it is. Oh my gosh, I get above negative 55 and I get an action potential. So now I've got an action potential that it's going to go down the axon. I really like this picture. I think it's a really good way of representing how a graded potential can fizzle or a graded potential can start an action potential. Okay, I think that's very clever of them. All right, so that's going to be it for the compare and contrast on local versus action potentials. So I'm going to stop this recording and you can move on to the next one.